Okay, so we are going to delve into the topic of filtration, which in a previous lecture, I used the analogy of a Finding Nemo fish tank to kind of diagram or describe what filtration is doing. Here I have another image that I want to go through that kind of shows you, what filtration is all about. So in this image here, imagine that I take some water and it's got a bunch of sand and silt and other large particulates. To filter it out means to take that mixture and put it over some sort of filter, such as filter paper. If you've ever seen filter paper, it looks just kind of like maybe softer but traditional paper. And that filter paper is going to capture all the big stuff while the small stuff moves through and is collected down at the bottom. And we call the fluid that is collected after something has gone through filtration, we call that filtrate. Now, that's a basic overview of how filtration works. We do it in chemistry to purify stuff, or biochemistry. We actually do it in ecology sometimes when we're looking at like organisms that live in the mud. But here we're going to apply this concept to blood. So I have down here a nice picture of blood. And you can see all of the particulates that are in blood. So remember, blood is going to be, between 40 and 45% cellular elements, and then the other part is going to be water and things dissolved in water. And so what we want to do is we want to basically filter out the large stuff. And so our contraption is not filter paper, but rather the glomerulus and Bowman's capsule. So Bowman's capsule is almost like the funnel. And the glomerulus is sort of going to be like the filter paper that kind of creates, in this case, like gravity, so some sort of pressure to kind of push the fluid through while still retaining the larger things. So the first thing that we want to do is we want to take a look at what is in blood. So I'm going to go ahead and make a list here of what's in blood, and then we're going to take a look at what we want to keep out of the filtrate and what we're going to let pass through. So now that I've written out a list of what is in blood, let's go ahead and take a look at, what would be retained. The capillary system is going to have small holes in it that's going to allow small stuff through, where the big stuff is going to be retained. So I've actually kind of organized this list by size, where my biggest stuff is at the top and my smallest stuff is at the bottom. So the capillaries themselves are going to try to make sure that most of the cellular elements, if not all of the cellular elements, are going to be retained in the blood. There's simply no reason to filter them out, because the whole point of filtering stuff out is so that you have an opportunity to either recapture it or let it go down the drain. And there's no reason why you would want to let perfectly healthy red blood cells, white blood cells, or platelets go down the drain. So you actually want to keep your white blood cells, red blood cells, and platelets back. And in fact, if you start to see these things in your urine, it can be indicative of kidney damage. So that's bad. Okay, so the cellular elements are going to be retained. The plasma proteins represent kind of an interesting paradox here. They are typically the largest elements of our plasma. But plasma proteins themselves range in size. So there's big plasma proteins, and then there's really small ones that you might not even call proteins, like certain types of hormones are pretty small, bigger than some of our nutrients, but not necessarily big enough to be like a plasma protein, like let's say low density lipoprotein. So plasma proteins represent kind of an interesting threshold here, where some of the plasma proteins, the smaller ones in particular, probably are going to get through. And the larger ones are going to be retained through processes that we'll get into in a few minutes. Generally, everything that I am bracketing there is going to make it through and be in the filtrate. So it's going to be filtered out. So let's take a look at what that is. If we think about our 4 classes of biomolecules, we have glucose, amino acids, and nucleic acids, and also lipids. Lipids are hydrophobic and tend not to be dissolved in plasma, but rather hitch a ride on the plasma proteins. So we don't really consider free fatty acids because there's unlikely to be very many of them. So the ones of importance are going to be any sort of simple sugar, amino acids, and nucleic acids. Waste products, which include urea, uric acid, a couple different types of bilirubin, bicarbonate, etc. Those are also going to be small and therefore make it through the filtration system and be in Filtrate. And then a number of ions. So remember, we're taking blood, plasma, and we're filtering it out. So we have to think about, well, what are the dominant ions in plasma? And it's the dominant ions in extracellular fluid. Those are going to be sodium, and they're also going to be chloride. So sodium and chloride are the big ones. But there's also going to be a small amount of potassium, and there's going to be hydrogen ions as well as a small amount of calcium, phosphate groups, etc. So sodium and chloride is not the exhaustive list, although they are far and away the most common ions you will come across. And of course, our predominant substance is going to be water. Let's go ahead and talk about the layers that are involved in our filtration system in the renal corpuscle that allow us to retain our cellular elements and our larger plasma proteins. So first I want to show you an image of the renal corpuscle and an electron micrograph of the glomerulus, which I think is really fascinating here. So I'm going to go ahead and highlight as I walk you through this. This is our afferent arterial. You can see here's our network of capillaries, and they seem to be surrounded by some yellow thing. It turns out that yellow thing is actually going to be important. Then you'll notice that Bowman's capsule, which is actually just a layer of simple squamous epithelial cells here, this Bowman capsule, very thin, kind of envelopes around it before coming to a point. And that point is going to be right here. Now, hopefully you kind of see the analogy between the glass filtration system and Bowman's capsule. Now, before going any further, I just want to draw your attention to these because I just think it's so neat. These are electron micrographs of glomeruli, and they do kind of look like little brains or maybe even a collection of tightly wound ramen noodles. But you can just see how condensed and what a network that really is. That actually plays a role in their function. So this is another example of the structure-function relationship. The tight weaving of those capillaries is important to allowing filtration to happen, as we're going to see in a little bit. Let's take a look at another picture. So again, you can familiarize yourself with the anatomy of renal corpuscle. We have our afferent arterial here, and then we have our glomerulus. Now you'll see a better picture of what in the previous image was shown as kind of a yellow draping over the glomerulus. Here it's shown in lilac, and it has a name. It is called a podocyte. Those podocytes are going to be important. Otherwise, why would we have them? So what are podocytes? Podocytes are specialized cells that actually wrap themselves around the capillary, and the word podo means feet. And so anytime you have a cytoplasmic extension in a cell, so let's say this is my cell and there's a cytoplasmic extension like that, we often call them feet or pseudopods, sometimes fake feet. And so they get these cytoplasmic extension, which I'm using as my fingers, and they wrap themselves around and kind of do this. So what does that look like? Doesn't that kind of look like a sieve of some sort or some sort of filtration device maybe? Indeed. So podocytes are going to be another layer in the filtration setup here. So if we think about filtration, we can almost think of filtration in layers. And in fact, there's going to be 3 layers that we need to concern ourselves with. And podocytes are certainly one of them. So it's the fact that they take these cytoplasmic extensions and wrap them around and kind of do this. and they create little slits that stuff has to get through. That's going to create another sieve for us to kind of help keep the bigger things back. All right, I also want you to take a good look at the anatomy of Bowman's capsule here. I'm outlining it in orange. The idea is that fluid, I'm going to use pink for Filtrate, that fluid is going to kind of leak out through these little slits that are made in between the podocytes here. Now, as fluid leaks out through this process of filtration, that fluid is going to start pushing against these walls, right? And eventually that fluid should kind of get bent this way into the proximal tubule. But the fact that Bowman's capsule has crevices like this that are very difficult to get out of is actually also going to play a role in filtration, because it's not an easy path for that fluid to get out of Bowman's capsule. It really has to kind of work its way around and then through a pretty narrow exit point in the proximal tubule. And so that's going to play a role. So I just want you to be aware of that there. Even though this does not come up in this lecture, there's our juxtaglomerular apparatus. Let's take a look at the 3 layers of filtration. I mentioned the word sieve already, and I'm going to use it here again. We can almost think of the renal corpuscle, and particularly the glomerulus and the wrappings around the glomerulus, as being like layers of sieves. So here's kind of a traditional sieve that we might actually use in certain types of ecology, where we're like looking for organisms in the sand, small stuff, or stuff in the mud. As a marine ecologist, we would use these to kind of sample the deep ocean and pull them up, and basically the larger organisms would get caught on the wire mesh and all the small stuff would go through. But now you can actually put these guys in and create additional layers. And in the same way, there are really 3 layers to speak of in terms of filtration. And it starts with the glomerulus itself. Capillaries come in 3 forms: continuous, fenestrated, and sinusoids. The most common capillary style is the continuous one, and this is the one that we think about. The continuous capillary has very few openings, and if they do, they're very temporary and very small in those little clefts. Fenestrated, to be fenestrated means to poke holes. So a fenestrated capillary is one that almost looks like small Swiss cheese. And so you can see here there's actually been like holes that have been poked in there, where a sinusoid has really big purposeful holes. The glomerulus is fenestrated. So what does that tell you? It acts just like a sieve. Now, if you look at the size of those holes, think about what might get through there. Red blood cells? No. Red blood cells are going to be about this big. They're too big. White blood cells are likewise going to be too big, so there's a white blood cell. And even platelets are probably going to be too big. But pretty much all of plasma, including the plasma proteins, will probably make it through. So the fact that the glomerulus is made of fenestrated capillaries at least retains the cellular elements. So getting past our fenestrated capillaries then is going to be all of plasma, which includes our nutrients, our waste products, our ions, our water, and our plasma proteins. And our plasma proteins are not waste products, and they serve a function in our body, so we really do not want them to exit if we can help it. And so these 2 right here, podocytes and the basal lamina, are going to help retain the plasma proteins, at least most of them. The small stuff still gets through. And the kidney, or the nephron, does have the ability to actually reabsorb some of them, but not usually all of them. Which is why you can actually measure some of your hormones, even ones that are like made of super small proteins. You can measure them in your urine. Probably the best example of that is going to be human chorionic gonadotropin, which is used to test for pregnancy. So why can you measure it in your urine? Because trace amounts of plasma proteins get through regardless. But I will say the podocytes and the basal lamina are able to retain most of them. So I've already introduced the podocytes as separate cells that wrap around the capillaries, but I have yet to introduce the importance of the basal lamina. So let's first take another look at the podocytes, and then we'll talk about the basal lamina. This is another image showing a podocyte, and I think it's really fascinating. So what I'm going to do is I'm actually going to highlight where the podocyte cytoplasmic extensions are, and I'm going to do that in pink. So you can see there's an extension, there's an extension, there's an extension. And so hopefully you can see, wow, those are some tight fits. So where are what are called filtration slits? And so the filtration slits are going to be where fluid has an opportunity to exit the glomerulus and go into Bowman's capsule. And you can see here, they're pretty small. There's a filtration slit. So I love this image for showing you just how tightly woven those podocyte extensions really are. Okay, so let me go ahead and write down some summary information here. Okay, so go ahead and write this down, pause the video if needed. So I want to point out something that I think is really fascinating. There's been studies that actually look at the effect of podocytes on filtration. And what would happen if you were to take these podocytes and you were to really thicken them up and make it so that the filtration slits were almost impossible to get through? What do you guys think would happen to your glomerular filtration rate? Is anything getting through that? Not really, right? I mean, very little. So if your podocytes are actually too tight, then your glomerular filtration rate is too low. And it turns out that that's actually a thing, to have a glomerular filtration rate that's too low. Consider the opposite. Consider what might happen if your podocytes were kind of atrophied and withered and then the gaps got really big. What do you think you might see? Well, your ability to retain plasma proteins would be compromised, and so you would start to see protein in your urine that you would not expect. So when podocytes atrophy, a diagnostic marker is, in fact, protein in the urine. Okay, let's take a look at our last filtration sieve, and that is going to be the basal lamina. Okay, in this image, we see a cross-section of a glomerulus, and we also see the cross-section of the podocytes. So really, the podocyte extensions are kind of coming out, and somebody's taken like a knife and sliced them, so you're looking at the cross-section here. So these are this is a cross-sectional view of the podocytes. And you can see the filtration slits, which I drew previously in red, so I'll draw them in red here too, are going to be right there. Those are the filtration slits. And then we also have our holes in our capillary, that's the fenestration, those are going to be there. And then we have a layer in between. Now, if you remember anatomy, hopefully, you remember that anytime you have a layer of cells, an epithelial layer, and it's stuck to something else, there's always some sort of basal lamina, which involves collagen fibers. The basal lamina here is going to act like gauze. And the gauze is actually a network of collagen. So collagen is a thick fibrous protein. Kind of looks like this, so you could almost imagine there's one layer of collagen and mesh, and then we're going to put another layer on top of that, and another layer on top of that, and another layer, and pretty soon you can see it's going to be really hard for stuff to get through that network. And so the basal lamina with the filtration slits is really important in retaining most of the plasma proteins, because a plasma protein, in order to get through, would have to actually try to find its way through one of those, a bunch of little tiny slits. And that's a tall order. So if you have a plasma protein, and I'm just going to put one right there, plasma proteins are not that big. But once again, you've got multiple layers here, layers upon layer. And the most likely course of action is that even if that plasma protein is capable of getting out here, it runs into this and is likely to get all tangled up and eventually find its way back in. And even if it does kind of get there, what if it gets trapped right here? Then again, it might bounce around a little bit and eventually find its way back in. There are some other things that are happening on the podocytes in particular that are kind of repelling the plasma proteins that are beyond the scope of what I talk about. But I do just want to put out that there's some other things going on that really help retain the plasma proteins. The end result, the basal lamina and the podocytes together, is such that we retain most of our plasma proteins. For my test in particular, I tend to treat this as we retain all plasma proteins. I don't consider the small ones that do kind of sneak through. So again, if you're thinking about it from my test perspective, go ahead and just write down that plasma proteins are retained with the basal lamina and the podocytes. So the next lecture is going to pick up with the gradient and how do we establish the gradient to allow filtration to happen. I'll see you soon.